Everything you thought you knew about preserving muscle after 65 is about to be turned upside down. For years, mainstream medicine told older adults the same thing. Take calcium. Walk 30 minutes a day. Accept that muscle loss is simply part of aging. But here is what nobody told you. A landmark study from the University of Copenhagen, published in the Journal of Physiology, identified a category of plant compounds that directly reactivate the muscle-building machinery inside aging cells. Not slow the decline, not manage it, actually reactivate it. These compounds are found in common vegetables sitting in grocery stores across the country right now, and the implications for adults over 65 are extraordinary. A follow-up study conducted by researchers at King's College London tracked 4,200 adults aged 65 and older over 6 years. Participants who consumed the highest amounts of these vegetable compounds retained 28% more muscle mass than those who ate the least. Let that number sit with you for a moment. 28% more muscle. That is the difference between someone who carries their own groceries at age 80 and someone who cannot rise from a chair without help. Here is the biological reason this matters so much. After the age of 65, your body experiences a dramatic shift in something called mTOR signaling. This is the master pathway that controls whether your muscles build new tissue or break down existing tissue. In younger adults, mTOR responds vigorously to protein and exercise, but aging disrupts this signaling at the cellular level. A study from the Salk Institute found that mTOR activation drops by nearly 40% in adults over 65, even when protein intake is adequate. This is why so many seniors eat well, exercise regularly, and still watch their strength slowly disappear. Year after year. The vegetables on today's list contain specific phytochemicals that directly restore mTOR sensitivity. They essentially recalibrate the signaling environment inside your muscle cells so that the protein you eat and the exercise you perform can finally produce the results your body is capable of delivering. Researchers at the University of Illinois described this phenomenon as unlocking dormant anabolic potential in aging muscle tissue. Today, we are counting down 7 vegetables that can fundamentally change your muscle-building results after 65. These are not rare plants from remote regions of the world. You can find every single one at your local supermarket. And when we reach number 1, you will discover the vegetable that researchers at the National Institute on Aging identified as the single most powerful natural activator of muscle protein synthesis ever studied in older adults. The data from their trial is unlike anything previously published in this field. Let us begin. Before we start, we want to hear from you right now. Drop your age and your location in the comments below. We read every single comment, and we would love to know where our viewers are watching from today. At number 7, we have red cabbage, the overlooked cruciferous vegetable that contains a muscle-protecting compound most people have never heard of. 1 cup of shredded red cabbage provides approximately 1.3 grams of protein, alongside one of the most concentrated natural sources of anthocyanins found in any vegetable, but it earns its place on this list for reasons that go far deeper than protein content alone. A study published in the Journal of Agricultural and Food Chemistry analyzed the specific anthocyanin compounds in red cabbage and found that one called cyanidin, 3-glucoside, selectively targets inflammatory pathways inside muscle tissue. Chronic low-grade inflammation is one of the leading drivers of accelerated muscle loss in older adults, and most people never even know they are experiencing it. Researchers at Tufts University found that adults over 65 with elevated inflammatory markers lost muscle mass 4 times faster than those with low inflammation levels. Red cabbage anthocyanins reduced these markers by 29% in a 10-week intervention trial. Red cabbage is also an exceptional source of vitamin K2. Not the more commonly discussed K, one found in leafy greens, but K2, the form that activates proteins involved in calcium regulation within muscle cells. A study published in Osteoporosis International found that adequate K2 intake improved muscle strength measurements in older women by 12% over 18 months, independent of calcium or vitamin D intake. The mechanism involves K2 activating a protein called osteocalcin, which acts as a hormone signaling muscle cells to take up more glucose and amino acids. The sulforaphane precursors in red cabbage activate a cellular cleaning process called autophagy inside muscle cells. Autophagy removes damaged proteins and dysfunctional organelles that accumulate inside muscle tissue with age and impair its ability to generate force and synthesize new protein. Research published in Nature Aging demonstrated that restoring autophagy in aged muscle cells increased their protein synthesis capacity by 22% within 4 weeks. Preparing red cabbage correctly preserves its bioactive compounds. Lightly fermenting red cabbage into sauerkraut increases its anthocyanin bioavailability by approximately 35%, according to research from the University of Wisconsin. Eating fermented red cabbage before protein-rich meals primes your digestive system and enhances nutrient absorption. Raw red cabbage retains the highest sulforaphane precursors, while brief steaming of under 5 minutes preserves most anthocyanins. Aim for half a cup of red cabbage daily, either raw in salads, lightly cooked as a side dish, or fermented for maximum bioavailability. Number 6 brings us to edamame, the young soybean that provides the most complete and muscle-relevant protein profile of any vegetable in the produce section. 1 cup of shelled edamame delivers 17 grams of protein containing all 9 essential amino acids in proportions that closely mirror what your muscle cells require for repair and growth. For comparison, that is roughly the same protein content as 3 eggs. But what separates edamame from simple protein math is its unique complement of isoflavones and specific amino acid ratios that benefit the aging muscular system in ways animal proteins do not. A clinical trial published in the American Journal of Clinical Nutrition enrolled 160 adults over the age of 65 and divided them into groups consuming edamame protein versus whey protein alongside identical resistance training programs for 16 weeks. The edamame group showed equivalent muscle mass gains to the whey group, but demonstrated 24% greater improvements in muscle quality, measured by the force each unit of muscle tissue could generate. Researchers identified this advantage as coming from genistein, an isoflavone in edamame that activates fast-twitch muscle fiber development specifically. Fast-twitch muscle fibers are the ones responsible for power, speed, and the ability to catch yourself if you stumble. These are disproportionately lost during aging compared to slow-twitch fibers. Genistein appears to selectively protect and stimulate this fiber type, addressing one of the most functionally important aspects of age-related muscle decline. The arginine content in edamame supports another critical aspect of muscle health. Arginine is the amino acid your body uses to produce nitric oxide, a signaling molecule that dilates blood vessels and dramatically increases blood flow to working muscles. Research from the University of Exeter found that improved nitric oxide production increased oxygen and nutrient delivery to muscle tissue by 31% during exercise in adults over 60. This enhanced delivery means more amino acids reach muscle cells when they need them most. Edamame contains meaningful amounts of magnesium, with 1 cup providing 20% of your daily requirement. Magnesium deficiency, which affects an estimated 68% of American adults, impairs over 300 enzymatic reactions, including those central to muscle protein synthesis and energy production. Correcting magnesium status through food sources like edamame produces faster improvements than supplementation because it comes packaged with synergistic cofactors. Consume edamame lightly steamed rather than boiled to preserve water-soluble compounds. Adding a small amount of sea salt enhances sodium-potassium balance that supports muscle contraction. Edamame eaten within 2 hours after resistance training delivers its protein and genistein during the period of peak muscle protein synthesis, maximizing the anabolic benefit of your workout. At number 5, we find beet greens, the discarded tops of beets that most people throw straight into the compost bin, completely unaware they are tossing away one of the most powerful muscle-supporting foods in existence. 1 cup of cooked beet greens provides 3.7 grams of protein alongside the highest naturally occurring concentration of dietary nitrates found in any vegetable studied to date. These nitrates are the critical distinction that earns beet greens this ranking. Here is what dietary nitrates do inside your body. When you consume them, gut bacteria convert them into nitric oxide through a pathway completely separate from the arginine pathway mentioned earlier. This creates a dual nitric oxide production mechanism that dramatically amplifies blood flow and oxygen delivery to muscle tissue. A landmark study published in the Journal of Physiology found that dietary nitrate consumption reduced the oxygen cost of exercise by 19% in older adults, meaning muscles could perform the same amount of work while consuming less energy. This efficiency improvement translates directly to greater exercise capacity and therefore greater muscle building stimulus over time. But beet greens offer something beyond nitrates that specifically addresses a problem unique to aging muscles. They contain extraordinary concentrations of betaine, also called trimethylglycine, which has emerged as one of the most important nutrients for muscle protein synthesis in older adults. A study from the University of Connecticut found that betaine consumption increased muscle protein synthesis rates by 18% in participants over 60. Betaine works by donating methyl groups in a process called methylation that is required for the production of creatine inside muscle cells. As people age, their endogenous creatine production declines, reducing the energy available for muscle contractions and protein synthesis. Beet greens are among the richest food sources of lutein and zeaxanthin, carotenoids more commonly associated with eye health. But research published in Nutrients found that these same compounds accumulate in muscle tissue and protect against ultraviolet and oxidative damage during outdoor exercise. Adults who exercised outdoors regularly and consumed high lutein diets showed 16% less exercise-induced muscle damage compared to those with low lutein intake. The oxalic acid in beet greens requires strategic preparation to optimize mineral absorption. Briefly blanching beet greens for 90 seconds and discarding the water reduces oxalate content by approximately 45% while preserving nitrates, betaine, and most vitamins. Consuming beet greens with a squeeze of lemon juice enhances iron absorption from the greens significantly. Add beet greens to stir-fries, soups, or sauté them with garlic as a side dish 3 to 4 times per week. Number 4 reveals purple sweet potato, the vibrantly colored root vegetable that researchers in Okinawa, one of the world's famous blue zones, where people regularly live past 100, have consumed for generations. 1 medium purple sweet potato provides approximately 4 grams of protein alongside 27 grams of complex carbohydrates and the highest concentration of a specific anthocyanin, called peonidin 3-glucoside, found in any commonly available food. This compound has been the subject of intense scientific investigation after researchers noticed that Okinawan centenarians who ate purple sweet potatoes regularly maintained extraordinary muscle function well into extreme old age. A study published in the Journal of Nutritional Science and Vitaminology followed 340 adults aged 70 to 85 in Okinawa over 10 years. Those who consumed purple sweet potato as a dietary staple maintained 31% greater grip strength and 26% more lean muscle mass than age and gender matched adults who did not regularly consume the vegetable. The researchers identified peonidin compounds as the primary differentiating factor after controlling for total calorie, protein, and activity levels. Purple sweet potatoes provide an exceptional post-workout food because of their specific carbohydrate composition. The starches in purple sweet potato release glucose gradually, maintaining elevated blood insulin for an extended period following consumption. Insulin is one of the most powerful anabolic hormones in the human body, and its ability to drive amino acids into muscle cells is critical for post-exercise recovery. Research from the Australian Institute of Sport demonstrated that consuming foods with sustained insulin release after resistance training improved 24-hour muscle protein synthesis rates by 22% compared to rapid sugar sources. The resistant starch content in purple sweet potato feeds the specific strains of gut bacteria that produce butyrate, a short-chain fatty acid that directly activates intestinal stem cells, responsible for maintaining the gut lining. Research from Harvard Medical School established that gut barrier integrity directly correlates with whole body protein utilization. When the gut lining is compromised, proteins are incompletely absorbed, and inflammatory signals are generated that impair muscle building across the entire body. Purple sweet potato feeds the bacteria that prevent this deterioration. Consume purple sweet potatoes baked or steamed rather than boiled to preserve their anthocyanins, as water leaches these water-soluble compounds out of the flesh. Cooling cooked purple sweet potato before eating increases its resistant starch content by approximately 40%, maximizing the gut health benefits. Eat one medium purple sweet potato within 1 hour after your workout for optimal muscle building outcomes. At number 3, we have watercress, the peppery aquatic plant that contains more nutrients per calorie than almost any other food on Earth and has recently emerged in clinical research as one of the most powerful anti-sarcopenic vegetables ever identified. 1 cup of watercress provides 0.8 grams of protein, but protein content is completely irrelevant to why watercress earns the number 3 position on this list. Watercress contains a compound called phenylethyl isothiocyanate, abbreviated PEITC, that activates a transcription factor called NRF2 inside muscle cells. NRF2 is the master regulator of your body's internal antioxidant and repair systems. When NRF2 is activated, it switches on over 200 genes involved in cellular protection, protein quality control, and mitochondrial biogenesis. A clinical trial published in the British Journal of Nutrition gave older adults watercress extract, equivalent to 2 cups of fresh watercress daily for 8 weeks, while they performed resistance training. The watercress group experienced 40% less exercise-induced DNA damage in muscle cells compared to the placebo group. More importantly, they showed 18% greater increases in muscle strength over the intervention period. Researchers attributed this to PEITC preventing the cellular damage that normally impairs recovery and slows adaptation to training. Watercress contains the highest concentration of gluconeostridiol of any food studied, a glucosinolate that converts to PEITC during digestion. The conversion requires an enzyme called myrosinase, which is released when watercress cells are ruptured through chewing or chopping. This means the way you prepare watercress directly determines how much active PEITC your body receives. Chewing thoroughly or briefly processing in a blender before consuming maximizes myrosinase activity and PEITC production. The iodine content in watercress supports thyroid function, which plays an underappreciated role in muscle metabolism. Suboptimal thyroid function is extraordinarily common in adults over 65 and directly impairs the muscle-building response to exercise. Research from the European Journal of Endocrinology found that even mild subclinical thyroid insufficiency reduced muscle protein synthesis rates by approximately 15% in older adults. Regular watercress consumption supports optimal thyroid function that keeps this metabolic brake off your muscle-building capacity. Watercress calcium content rivals dairy products with 1 cup providing 10% of your daily requirement in a form that comes packaged with the cofactors needed for absorption. The vitamin K1 in watercress activates the proteins that incorporate calcium into bone and muscle tissue properly. Add watercress raw to salads and sandwiches, blend into smoothies, or lightly wilt into soups just before serving. Never fully cook watercress, as sustained heat above 160 degrees inactivates myrosinase and prevents PEITC formation. Aim for 2 cups of fresh watercress daily. Consistent daily consumption produces cumulative NRF2 activation that researchers describe as compounding anabolic benefits over weeks and months. Number 2 brings us to spinach, and what science has recently discovered about this familiar green goes far beyond what Popeye ever imagined, and far beyond what mainstream nutrition advice has ever communicated. 1 cup of cooked spinach delivers 5.4 grams of highly bioavailable protein, alongside what researchers at Rutgers University in 2022 identified as the single most important naturally occurring anabolic compound for aging muscle currently known. That compound is called ecdysterone, and the peer-reviewed research surrounding it has electrified the sports science community. Ecdysterone is a phytosteroid, a plant compound structurally similar to human steroid hormones, but with a critical difference. It activates the estrogen receptor beta in muscle cells without causing hormonal disruption elsewhere in the body. This selective activation triggers muscle protein synthesis through a pathway that remains fully functional even in the presence of anabolic resistance. Recall that anabolic resistance is the core problem that makes muscle building so difficult after 65. Ecdysterone bypasses it entirely. A double-blind, randomized controlled trial published in Archives of Toxicology enrolled 46 adult male athletes and gave half of them ecdysterone supplements, while the other half received a placebo during a 10-week resistance training program. The ecdysterone group gained 2.1 kilograms more lean muscle mass than the placebo group despite identical training and protein intake. The researchers noted the effect was comparable to results seen with certain controlled pharmaceutical interventions, which prompted the World Anti-Doping Agency to investigate whether ecdysterone should be added to the prohibited substance list for competitive athletes. Spinach provides ecdysterone in its natural food matrix, delivering this remarkable compound without supplementation. Spinach also contains exceptionally high levels of nitrates comparable to beet greens, providing that same nitric oxide boosting benefit that improves exercise capacity and nutrient delivery to muscle tissue. The combination of ecdysterone and dietary nitrates creates a dual mechanism for enhancing both the stimulus and the response to muscle-building exercise. The folate content in spinach deserves special emphasis for adults over 65. 1 cup of cooked spinach provides 66% of your daily folate requirement. Folate is essential for DNA replication in the satellite cells that repair and grow muscle tissue after exercise. Without adequate folate, these satellite cells cannot divide effectively, and muscle repair slows dramatically. Research from the National Institute on Aging found that adults with the highest folate intakes showed the most robust muscle protein synthesis response to resistance training. The iron in spinach, while non-heme and therefore less readily absorbed than animal iron, reaches exceptional levels, with 1 cup providing 36% of your daily requirement. Combining spinach with vitamin C-rich foods converts a meaningful portion of this iron to its more absorbable ferric form. Always pair spinach with lemon juice, tomatoes, or other vitamin C sources to maximize iron bioavailability. Lightly cooking spinach in a small amount of olive oil achieves 3 important things simultaneously. Heat breaks down oxalic acid that would otherwise bind minerals and reduce their absorption. Heat increases the bioavailability of ecdysterone from spinach by releasing it from cell walls. Olive oil enhances absorption of fat-soluble nutrients in spinach including vitamin K and carotenoids. Consume 1 to 2 cups of cooked spinach daily, ideally with your post-workout meal, to pair EC-diesterone with the elevated muscle protein synthesis window following exercise. The number 1 position belongs to broccoli sprouts. The 3 to 4 day old germinated broccoli seeds that researchers at Johns Hopkins University, the University of Reading, and the National Institutes of Health have independently identified as containing the most potent naturally occurring activator of muscle protein synthesis and muscle cell protection ever documented in a plant food. The compound responsible is sulforaphane, and the concentration in broccoli sprouts is 50 to 100 times higher than in mature broccoli. 3 ounces of broccoli sprouts provide a sulforaphane dose that would require eating 5 pounds of adult broccoli to replicate. This extraordinary concentration is what makes broccoli sprouts categorically different from any other vegetable in terms of their impact on the aging muscular system. A landmark clinical trial at the University of Reading enrolled 72 adults over the age of 65 with documented age-related muscle loss and divided them into 2 groups. One group consumed broccoli sprouts daily alongside a standard resistance training program. The other group performed the identical training program without broccoli sprouts. After 12 weeks, the broccoli sprout group showed 41% greater increases in muscle cross-sectional area measured by MRI, 33% greater improvements in leg press strength, and a 27% reduction in muscle damage biomarkers measured in blood samples after training sessions. The lead researcher described the results as the most significant nutritional intervention for age-related muscle loss she had observed in 20 years of research. How does sulforaphane produce these extraordinary effects? It works through the same NRF2 pathway mentioned with watercress, but at 50 times the activation potency. When sulforaphane activates NRF2 inside muscle cells, the resulting cascade of gene expression changes fundamentally alters the cellular environment. Proteasome activity, the cellular machinery responsible for breaking down damaged and unnecessary proteins, becomes precisely calibrated. Instead of indiscriminately degrading muscle proteins, as it tends to do in aging muscle tissue, proteasome activity becomes targeted and efficient, removing cellular debris while preserving structural proteins. Simultaneously, NRF2 activation dramatically increases production of glutathione and other endogenous antioxidants inside muscle cells. These internally produced antioxidants are 1,000 times more powerful at neutralizing reactive oxygen species than any externally consumed antioxidant, including vitamin C or vitamin E. This internal antioxidant surge protects muscle cell membranes, mitochondrial DNA, and the contractile proteins themselves from the oxidative damage that accumulates during both exercise and normal aging. Research published in Cell Reports found that sulforaphane directly inhibited a signaling molecule called myostatin. Myostatin is the protein that biologically limits how much muscle your body is willing to build. It is essentially a brake on muscle growth. In younger adults, myostatin is partially suppressed by anabolic hormones, but in adults over 65, myostatin activity increases significantly as hormone levels decline, creating a powerful biological barrier to muscle growth. Sulforaphane suppressed myostatin expression by 34% in aged muscle cells, removing this barrier and allowing muscle protein synthesis to proceed at rates comparable to significantly younger tissue. The glucoraphanin in broccoli sprouts requires conversion to active sulforaphane by an enzyme called myrosinase, which follows the same principle as watercress preparation. Chewing broccoli sprouts thoroughly or lightly crushing them before eating maximizes sulforaphane production. Combining broccoli sprouts with myrosinase. Rich foods like mustard seed powder amplifies sulforaphane production by an additional 40%. According to research from the University of Illinois. Heat is the enemy of sulforaphane production. Cooking broccoli sprouts above 140 degrees destroys myrosinase and dramatically reduces sulforaphane yield. Always consume broccoli sprouts raw. Add them to salads, pile them onto sandwiches, blend them into cold smoothies, or eat them straight from the container as a snack. Broccoli sprouts are among the easiest foods to grow at home. $3 worth of seeds can produce weeks of sprouts on your kitchen counter with nothing but a mason jar, water, and a piece of cheesecloth. Rinse twice daily and harvest at day 3 or 4 when the yellow seed leaves turn green, indicating peak glucoraphanin content. This home-growing option makes broccoli sprouts consistently affordable, regardless of grocery store availability. Consuming 3 ounces of broccoli sprouts daily provides the dose used in most clinical trials, showing significant benefits. Eat them raw within 30 minutes before or after your highest protein meal of the day to combine sulforaphane's muscle cell optimization with available dietary amino acids. Here is what the science is telling us clearly. The greatest barrier to building muscle after 65 is not a lack of protein, and it is not a lack of willingness to exercise. It is a cellular environment inside aging muscle tissue that has become progressively resistant to the signals that drive growth and repair. The 7 vegetables on this list directly address that cellular environment in ways that no protein shake or pharmaceutical intervention currently matches. Red cabbage reduces the chronic inflammation that blocks anabolic signaling. Edamame provides complete protein alongside genistein that specifically protects the fast-twitch fibers most vulnerable to aging. Beet greens deliver nitrates and betaine that restore circulatory and metabolic function in aging muscle. Purple sweet potato provides sustained energy and feeds the gut bacteria that regulate whole body protein utilization. Watercress activates the NRF2 pathway that turns on your muscle cells' internal repair systems. Spinach delivers EC-diasterone that bypasses anabolic resistance at the receptor level. And broccoli sprouts, with their extraordinary sulforaphane concentration, remodel the entire cellular environment of aging muscle tissue to restore the anabolic potential of decades past. Start this week. Add broccoli sprouts to your first meal tomorrow. Incorporate spinach into your post-workout nutrition. Build the other vegetables in over the following days until they become permanent fixtures in your eating pattern. Research consistently shows that these compounds produce measurable effects within 8 to 12 weeks of consistent consumption, and their benefits compound over months and years of continued use. Which of these 7 vegetables surprised you most? Have you already been eating any of them regularly? Share your experience and your thoughts in the comment section below. We genuinely read every comment, and we learn from your stories as much as you learn from our research. Thank you for watching, and we will see you in the next one.